Okay, thanks to organizer to allow this symposium. The first slide is to remember Fernando Lopez da Silva, who was really a referent person in the field of neurophysiology, EEG, magnetoencephalography, and epilepsy. He passed away this year in May, and we are indebted with him for many, many years of guidance in this field. Next slide, please. Or maybe. Okay, so just to roll. Okay, I will represent in this talk the core contents and the main conclusions of our work group. You see the authors of this position paper of the International Federation of Clinical Neurophysiology. The focus was on the frequency and the topographic analysis of a resting state EEG RIMS. I have no conflict of interest. The main issue for the clinical research is to test the properties, the qualification of biomarkers for the diagnosis of monitoring of therapy response. And concerning the diagnosis, the gold standard is the ability to perceive a disease by means of biomarkers in the very early stages of the disease, when you have very mild symptoms, or ideally no overt symptoms, but just changes in the pathophysiology of the body. And to follow the progression or evolution or changes in the disease over time, up to the most severe manifestations of the disease. The resting state EEG in clinical research was introduced a lot of years ago. Here you see an inset of the Sistine Chapel in Rome, so you can visit this, from Michelangelo. And you see it's very famous, in which God touches the first man, Adam. And a researcher several years ago, Meshberger, in a very important medical journal, JAMA, published a hypothesis that Michelangelo drew a brain in the mantle of God, and you can recognize something similar to a brain stem. And the concept was that Michelangelo wanted to represent the importance of brain to realize cognition, representation, perceptions. And you see that there are some similarities to typical representations of the brain in the mammals. And you see the cerebral cortex as some similar feature. And the concept is that the resting state EEG represents a neurophysiological system maintaining brain arousal and vigilance over time. And so the concept is that there is some ascending reticular activating systems, for example, dopaminergic and cholinergic, but also noradrenergic and serotoninergic and so on, that can maintain the proper cortical tone, the proper brain arousal, and modulate the vigilance in the best way as a pillar of cognition, memorization, reasoning, and so on. And we know that this ascending activating reticular system is able to modulate thalamus and cerebral cortex, in the generation of EEG RINs, in the resting state EEG RINs, and to regulate vigilance. Here you have from the courtesy by Professor Crunelli a nice representation of the cerebral cortex and the thalamus, the thalamic relay nucleus, for example, and also to nucleus reticularis thalamic. And you see the typical neurophysiological circuit at the basis of EEG RINs in the resting state condition and vigilance. You see the cortical pyramidal neurons and the fibers that can modulate the activity of the thalamocortical neurons. And you see that the cortical pyramidal neurons and the fibers can modulate relay thalamic neurons, but also reticular neurons of the thalamus. And the same is true in the opposite direction. The corticothalamic neurons have fibers modulating cortical pyramidal neurons and also reticular neurons in the thalamus. So in these circuits, you can measure in vivo and in vitro resting state EEG. And you see typical oscillations at 10 Hz, 10 cycles per second of EEG during quiet wakefulness. And very similar EEG oscillations can be recorded with electrodes put in the thalamus. And you see now is in mice these recordings from Crunelli, but the same is true for humans. And you can see that you have a lot of similarities between the alpha RIMS, 10 Hz oscillations during ongoing and quiet wakefulness in cerebral cortex and in the thalamus. And there is a relation with action potentials in the corticothalamic neurons. And you see that there is a very clear relationship between the negative and positive peaks of alpha oscillations and the trains of action potentials in the corticothalamic and thalamocortical neurons. So the challenges for the recording the analysis of resting state EEG for clinical applications are the number of electrodes to be used to record the resting state EEG, and also how to record EEG concerning the instructions to the subjects and the quiet wakefulness conditions, and also if the analysis should be focused on cortical sources or at sensors level, if when and how we can use linear and nonlinear measurements, and if our outcome, our results can be used for diagnostic purpose or monitoring of the diseases, so it's just a picture of human neurophysiology. So a main issue to localize the sources of EEG are the fact that you have had volume conduction effect. And so, for example, in this picture, you can see that, for example, the activation of the sulcus in the cerebral cortex can generate EEG signals far from the source. So if you just analyze the scalp electrodes, you can see a signal, EEG signal here, but there is no source underlying the electrode in this case. In other cases, you have a mix. For example, in the case of the left part of the picture, you see that these cortical sources and these cortical sources can generate a signal that you can record in B and in A. So there is the problem of volume conduction. And to analyze the data and obtain functional connectivity measurements, you have additional challenges. For example, in this case, you see that when you have this kind of activation of in a source, you have EEG signals in A and in B, and you can obtain some coherence between the EEG signals in A and B. But this is a fake coherence, of course, because there is just one source. And the same is true in this case, in which, for example, in the real case, you have two sources, one tangential here and another radial here. And if there is some real, true, functional connectivity between these sources, when you see the projection of this source in B and the projection of this source in C, you have a coherence in B and C, which don't correspond to the connectivity between the sources. So this is really tricky. And the same is true with the concept of the cascade flow effect. For example, if you have these sources and the directionality of the true connectivity with these sources and directionality with this source, you, at the scalp level, could see also this fake connectivity. And you don't have this connectivity in the true sources. And so you really in the trouble when you analyze the EEG at the scalp level and you want to obtain some representation of the cortical connectivity. Here you have a picture of some concepts concerning the use in clinical research of resting state EEG. For example, we recommend, in line with the International Federation Clinical Neurophysiology Guideline on the montages from Professor Sick and colleagues, we recommend the use of high resolution in the EEG from now we have several systems, 128 or 256 channels. But we, in this our position paper, accepted the retrospective study on archive EEG recorded with standard clinical electrode montage 1020 with 19 and 21 electrodes, but just for retrospective meta-analysis and with results to be validated in prospective high-resolution EEG studies. And then we propose different conditions of resting state, and the first is to test the ability of the brain to maintain eyes closed condition or eyes open condition with 3-5 minutes of eyes closed and 3-5 minutes of eyes open, or to check the ability to switch on and off with the resting state with alternated up open eyes and open close in 30 seconds, and then to study the transition from resting state to sleep, also for some analysis of epileptiform activity. And so a long recording of 30 minutes in the resting state eyes closed to go to the non-REM condition. Concerning the EEG source analysis, we recommended the use of open source platforms so the people can replicate your results, but we didn't give any specific indication for Loretta or OHBM or other platforms because there is the lack of comparison of results in the literature. You see here, that we recommend also for clinical applications the correlation with cerebrospinal fluid or blood biomarkers when appropriate, and the correlation with the neuroimaging data, structural and functional. Concerning the frequency bands, you see we revealed that we could not give final recommendations because we have different indications for frequency bands, alpha, beta, gamma, theta, and delta. You see different guidelines, even from International Federation of Clinical Neurophysiology, with slightly different frequency bands. And we also explained in the manuscript that we have some slowing of alpha frequency in several brain diseases, so we recommended the use on individual basis of alpha band analysis in the resting state EEG based on the work by Professor Klimesh. Concerning the EEG source analysis, we revised the literature and see good consistency of the results of EEG source analysis and estimation with different platforms, and so we think that it's appropriate for clinical research. We observe some inconsistencies and some variability in the literature when you compare different platforms for EEG source connectivity. So we think that we need more basic research before clinical practice with EEG source connectivity. And also concerning linearity and non-linearity, we observed that linear features are used consistently in different papers, and non-linear measurements need more research before the application in clinical practice. So I have no time for show you some applications, but just to see that we concluded our paper recommending cooperation with the Human Brain Mapping Organization and the Alliance Against Epilepsy to have some global initiative for the validation of these procedures. Thanks for your attention.